The overall goal of this procedure is to differentiate mouse embryonic stem cells into motor neurons. First, culture mouse embryonic stem cells on primary mouse embryonic fibroblasts and supplement with leukemia inhibitory factor. Then, enhance the neuralization process by adding noggin, BFGF, and FGF8. Induce motor neuron specification by incubation with retinoic acid and smoothened agonist, which is followed by axon elongation and motor neuron maturation. Finally, use immunofluorescence microscopy to examine differentiated MES cells that express strong GFP fluorescence.
Generating large quantities of motor neuron facilitates research in motor neuron diseases. Compared to current existing protocols, this approach of differentiating mouse embryonic stem cells into motor neurons is more efficient.
This method can provide insight into spinal muscular atrophy. It can also be applied to other motor neuron diseases such as amyotrophic lateral sclerosis.
To plate primary embryonic mouse fibroblasts, coat 4 100 mm tissue culture dishes with 8 ml of 0.1% gelatin solution. After 30 minutes at room temperature, discard the excess liquid. Now, dilute 1 vial of mitomycin C inactivated PMEF into 40 ml of PMEF media, plate onto the 4 plates, and culture for up to 1 week. Next, defrost 1 vial of MES cells in a 37-degree water bath. Wash the cells with 5 ml of ES media in a 15 ml tube. After centrifuging at 800 RPM for 5 minutes, aspirate the supernatant and